Good afternoon, everyone. My name is Noor Sherif, and today my colleague Youssef Ashraf and I will be discussing genetic composition of polar bodies and Barr bodies. But before we start, I just want to review really quickly the process of gametogenesis. So, as you guys remember, from gametogonia to a primary gametocyte is through mitosis. And then from primary gametocyte till secondary gametocyte, that is through meiosis 1. And then after producing from secondary gametocyte till around spermatid or an ovum, that is through what? Meiosis 2. And here I'm going just to discuss quite confusion that has been the number of chromosomes and number of chromatids for gametogenesis in general. I just want you guys to focus on this picture before. Here we have 2 chromosomes. How do we know these are 2 chromosomes? It's because we have 2 centromeres. Although each one of them is only one single chromatid, we still classify them as 2 chromosomes each one. And here we have also 2 chromosomes, but instead of just one single chromatid, we have 2 sister chromatids. So representing total of what? 4 chromatids. So now we have 2 fates for an oogonium. We have it will either proliferate or differentiate. So if it will proliferate, it has to do interphase first, which is basically what? G1, S phase, and G2. And as you guys can see in here we have 2N and 2C. After it copies the DNA replication, here we have the single chromatid being replicated, each one of them. So we'll have another copy here, and the same applies for the blue one. So here we have 2 chromosomes, as I have mentioned before, but a copy of the chromatid. Okay. And then it will undergo mitosis. So it's basically going to be the same as this one, just a division of it. And the reason is because we wanted to proliferate and just have more cells of it. And now when it decides to differentiate, it will undergo again interphase, which I have already discussed. And then it will undergo meiosis 1. In meiosis 1, it's going to split the pair of the chromosomes into halves, as you guys can see. So the number of chromosomes and the number of chromatids are going to be halved. So instead of 2N, in here it's going to be 1N. And instead of 4C, each one of them is going to be 2C. And in meiosis 2, here we formed what? If we're talking about oogenesis, is it primary oocyte or still primary? That's correct. So now primary oocyte is going to undergo what? Meiosis 2, splitting the number of chromatids into half in each one of them. So now here we will have 1 chromosome and still 1 chromosome. So 1N, 1N. And in each one of them in here, it was 2N because 2 single chromatids. Now we have in each one of them 1 single chromatid forming 1C. I hope that was clear. And just to point out the fact here, we have evidence supporting from the book in our medical embryology book stating that from the beginning of the oogonium, it will have only X chromosomes. Till the end, it will still have the X chromosomes as it's provided here, that only one primary oocyte gives rise to 4 daughter cells, each with 22 plus 1X. Thank you, guys.
Okay. Good afternoon, everyone. I hope you're all doing well. So now with my part, this is basically my conclusions of the article. So after thoroughly going through the internet and books, I can safely conclude that a female only has an X and an X chromosome, no matter her chronological life, which is developmental life, so before birth or after birth, and anywhere in her body. So whether it is in her gonads or whether it is in her polar bodies. Supported from the article that the doctor was presenting, it clearly states that, and I quote, the sex chromosomes would be depicted as female XX, male XY. However, this figure 5, is actually talking about fertilization and sex determination, which actually is irrelevant to the topic of a female having a Y chromosome in her polar body. And the polar body, as we've said, its only function, primary function, is to decrease the number of chromosomes from diploid to haploid. Okay? Next up, this is the polar bodies. It was also mentioned in the article. Polar bodies, they're basically inactivated X chromosomes. They're randomly activated in the woman's cells. So just any cell you take, she has two X chromosomes, one of them is inactive. That inactive chromosome is called Barr body. The reason for Barr bodies, or the purpose, is to achieve dosage compensation. Dosage compensation is basically because a man, he has a Y chromosome, and that Y chromosome is non-functional basically. Its only function, or its primary function, is for depicting sex or gender. So if a woman has two X chromosomes that are functioning and showing genetic compositions and stuff, and a man only has one X chromosome showing and the Y isn't working, that would cause an imbalance in genetic compensation or genetic variation. So again, for the compensation, only a female has only one X chromosomes working. And an interesting fact, and I know that 3/4 of our batch, they have pictures of a cat in their profile pictures. So I'm guessing you like this one. Calico cats. Calico cats, as you can see, they're cats with many colors, usually 3, okay? And this is actually because of the Barr bodies. So, and it's usually in females, because as we said, in males, they only have one X chromosome. And in female, they have the Barr bodies. Okay, last part. So, Barr bodies, as we've said, so it's randomly depicted between cell and cell. So, for example, in this spot, one of the X chromosomes, they're active, which would depict the color beige. Another part would depict the color black. So one X chromosome has beige, the other one has black.